Hello everybody, this is Suman from MultiCASE and I welcome everybody and thanks for joining this webinar. Today I'll be talking on the topic of expert review in the ICH M7 guideline based risk assessment of mutagenic impurities. But the way I plan this talk is to talk a little bit differently. That is I'll be talking more about the type of evidence and the order of different types of evidence that are required during the expert review process. So without further delay, let's start. So this talk has a limited scope because of the limitation of time, and so I'll be specifically talking about the mutagenic impurity that are mutagenic due to DNA reactivity as indicated in the ICH M7 guideline. The objective would be to use the expert review process to achieve a clear positive or negative conclusion after doing the basic and default QSAR evaluation using a software. And other type of genotoxic endpoints like chromosomal aberration or micronucleus type of topics are out of scope. Just as you can understand that this small time is not enough for it. So now can we define by ourselves what could be an expert review in this particular context? So I think the expert review process for ICH M7 guideline based discussion of mutagenic impurities is an investigation and validation of computationally identified reasons of DNA reactivity of chemicals by a human expert. In this definition, few things stand out, and the first one is the reasons of DNA reactivity. So when I say reasons of DNA reactivity, I'm trying to indicate towards the identification of structural alerts in a chemical by a computer algorithm or a computer program, and this process should be done by a human expert. So with that definition in mind, we can move on, and it would actually also set a stage for an explanation of the upcoming slides. So this table is very known. I'm sure everybody has seen it sometime. So it shows different classes that are defined by the ICH M7 guideline for the impurities in pharmaceuticals and drugs. And the first three categories, class 1, 2, and 3, they are a positive class. That means they indicate the mutagenicity of the compound in question. And the last two, 4 and 5, are the negative impurity as a result of the whole process. And the first two classes, class 1 and class 2, are based on the availability and use of experimental data. So there is no exception to it. So in order to get to class 1 and class 2, we got to have experimental data for them, experimental data for mutagenicity and experimental data for clear carcinogenicity of those compounds for class 1. And for the class 2, the compound should be a known mutagen. The class 3 is a positive result as a result of QSAR application, and class 4 is a negative result as a result of the QSAR application. But the last class, the class 5, can be based on the experimental data as well as QSAR. So with that in mind, let's go on. So the ICH M7 guideline tells us that we can use two complementary QSAR methodologies to assess computationally the mutagenicity of drug impurities. And those two methodologies are a statistical based methodology and an expert rule based methodology. So that's the basic starting point. However, the guideline also includes a provision to apply expert knowledge, which can be used to evaluate the results from the QSAR methodologies. And the expert review, by definition, should be performed by a human expert who has the knowledge of the problem in hand, which I termed as domain knowledge, and he should have some experience of dealing with QSAR technology, as well as the problem of mutagenicity and carcinogenicity in impurities. So these are few points about the provision of expert review in ICH M7 guideline. Now, as far as the importance of expert review and judgment, I would like to emphasize the fact that expert review overall is a beneficial step to perform. I understand that sometimes it is a little bit cumbersome, time-taking, and tedious, but I would like to argue in this slide that nonetheless it is actually a very beneficial step that if we perform, we would save a lot of time, money, and resources later on. So in the immediate benefits of expert review, we can use expert review to verify the accuracy of the results by considering additional evidence. We can increase the confidence of the software predictions using expert review. We can also resolve different types of conflicting outcomes. For example, if we get a positive outcome from the statistical system and negative from a rule-based system, we can use expert review to resolve those conflicts. And interestingly, expert review can also act as a buffer for different changes in the outcomes of software when we change the versions of the software. So as you might have already seen, that we keep on releasing different versions of the software and databases and models in every year or twice a year or something like that. And it can result into changes in the outcome for a single compound or particular number of compounds. So if we use expert review to evaluate the outcomes of software, more often you will see that the software changes result into outcome changes, but the outcomes from the expert review doesn't change that often. So that's a very good thing. In the similar line, if we use expert review, we can actually come to the same conclusion which we can get if we have applied different software. That is also has a very practical implication. Suppose somebody works in a particular pharmaceutical company and uses a software for a particular vendor. If he moves to a different company and uses a different software from a different vendor, the expert review can be very helpful to shield him from different outcomes. Other very important fact is that expert knowledge recently has been shown that it can overturn almost 26% of the predictions from the software, and majority of those predictions are actually downgrades. So downgrade means converting to negative from positive, or from positive to inconclusive, and so on. So it's a good thing. It also resolved almost 91% equivocal and 75% out of domain outcomes, and that's a very promising data as far as expert review is concerned. So it shows the incentive of using expert review. Also, in this, because of this kind of changes after the expert review, it plays a very important role in the decision-making workflows of regulatory agencies, and which can be seen in this particular paper that I cited here, which has been published very recently by the group of scientists from CEDAR FDA, and both of these data is last 2 points are coming directly from that paper. So in summary, expert review is very beneficial, and we have a lot of incentive to apply it. Now, there are a few challenges, of course, as I mentioned before, that sometimes expert review could be time consuming. It is tedious, of course. It needs expertise in the field of QSAR, genetic toxicology, analysis of impurities, and so on. It can sometimes also be subjective, and one of the things that we try to do during expert reviews to reduce this subjectivity by providing useful data and evidence while a person is performing the expert review. And of course, the expert review by definition cannot be automated by a software, but it can be assisted by the evidence generated by a software. So this is very important factor to keep in mind, that expert review is not an automated process by a software. Now, why we even need expert review? Don't we have model predictions coming out of a software system? So what is the difference between default model predictions, which are raw outcome from QSAR models, and the final expert decision? So let's talk a little bit about that. So default model predictions are generic in nature. Now, generic means they do not really care particularly about your query compound. The alerts that are present identified in your query compound, they have been found by analyzing a lot of chemicals from training set, or by a general knowledge of the expert, but they do not really focus completely on your query compound. And the important fact that should be kept in mind is that default model predictions are basically in order to identify structural reasons of mutagenicity. That is, the identification of alerts is the main objective of default model predictions. Now, these alerts that I'm talking about here, they are either learned from a collection of training compounds, in the case of statistical models, or they are based on general knowledge of mutagenic structural motifs by the experts. So they are, in essence, generic in nature. So that's where the distinction comes between the default model prediction and expert decision. The expert decisions, on the other hand, is specifically oriented towards the query structure that you are interested in. So in the case of expert decision, we check out, or we try to find out, if the identified alerts are valid for your query compound or not. And also, additional knowledge can be incorporated in the expert review process, and those are also specific towards your query chemical. For example, ADME parameters or information about your chemical, physical-chemical properties of a chemical is considered. So default model predictions are generic in nature. Their main goal is to identify alerts, but in the case of expert decision, we specifically focus on the query structure that is in question. Now, I tried to write few core principles that I have found during the last several years that plays important role in the expert review process. So if the decision is based on experimental data, like for example Ames test or carcinogenicity assay, it is important to determine the acceptability of the data. We'll talk a little bit more about this thing later on, but the acceptability of the data, if we are using it in our decision making process, it has to be determined. Now, if an alert is identified, which of course is done when the experimental data is not available, as we have seen in the previous table, then the objective of the expert review process is to determine the validity of that alert. That's a very important aspect of expert review. Suppose some unknown features or uncovered features are identified. An important objective of expert review is to determine the reactivity or the mutagenicity potential of those features. So I think these are the basic principles of expert review. There might be others, but these comes first in my mind when I'm thinking about expert review. Now, what are the basic ingredients before we go for an expert review? So prerequisites of an expert review process. So we need to have Ames data, experimental Ames data for the API. Sometimes if we can get our hands on carcinogenicity data, if available for the API, that is also has to be recorded and submitted as part of the process. Any experimental Ames data or carcinogen data, if available for the impurity, that should also be kept as part of the process. So the experimental data for both the API and, if available, if we are in good luck, for the impurity should be recorded. We have to get the QSAR predictions from the two methodologies, that is statistical and expert rule based methodology, before we go into doing the expert review. And in order to do the review process, to validate the importance of relevance of the alerts, we need to have a database of compounds with known experimental Ames data and underlying experimental details to do different types of activities, including analog search, which I'll be talking more in detail later on. So these are basic ingredients that we need to kept nearby before we go into do the expert review process. Okay, one thing that we should not forget before we even do any kind of prediction or use a software, that we need to pre-process the chemical structure that we are trying to evaluate. And what do we mean by chemical pre-processing? Another word for pre-processing is curation. So, for example, removal of inorganic salt parts. So, if you have a sodium salt of some drug, it is good to remove the sodium part, chloride, potassium salt. Those inorganic parts should be removed. Then you will be left with a charged organic part. So, it is good to neutralize the charges. Otherwise, what will happen that you are gonna get multiple out-of-domain predictions, or if already some experimental data is available in the software databases, you are not gonna get that if there is a charge present in many cases. Not always, but in many cases. If you are dealing with a mixture where there are different components in the single compound, those mixtures should be separated and then evaluated separately. And use of proper tautomeric form. So, as you know, in organic chemistry, a compound can be present in different forms, and each form look differently. They have a different structural representation, and it is important to use a proper tautomeric form in order to get proper results. And some of the factors that are relevant here is that if you use proper tautomeric form, then you will avoid missing any available experimental data in the database. So if you submit a wrong tautomer, wrong means a different tautomer which is already than what is already present in our database, obviously you are not going to get the experimental data from the database. Using proper tautomeric form also reduces chances of getting out-of-domain results. Or sometimes you can get an alert which is simply an artifact if we use a different tautomeric form. So in very near future, we are going to introduce a tool inside the data curator of QASE Ultra, our QSAR program, which will deal in a very nice way about all these tautomeric forms of the query compound that you are trying to submit to the software. And finally, if there are very small inorganic chemicals, like for example hydrochloric acid or single metal atoms, those compounds, those chemicals are not actually suitable for QSAR analysis. So instead of doing QSAR, maybe it is important to search exact hits or database to get any toxicity property for those chemicals. QSAR is not really relevant or gives good results for those chemicals. QSAR is more suitable for slightly drug-like organic chemicals. So what are the different software that we from MultiCASE build, develop and give to you, which are relevant in this expert review process? So to start with, our main software in this area is QASE Ultra, which is our main QSAR software. And we also obviously need databases. And for ICH M7 type of problem, we have two very important databases. One is a large database for bacterial mutagenicity and rodent carcinogen databases. These databases are very significant in size, and they are updated regularly, and we do manual curation as well as automated curation, and they are continuously improved year by year. So these are the databases that are required. And the QSAR models that we supply are, of course, both statistical model and expert rule based model. In statistical model, we have two different models, GT1B-mut and farm-B-mut. One is the main statistical model, and another one is an extended coverage statistical model. And of course, we have a very good expert rule based model called GT Expert. Now, for data pre-processing, which I just talked about, we have a very good tool inside Case Ultra called Data Curator that can be used for everything that I talked about, like removal of salts, finding and treating mixtures, neutralizing charge, and so on. We also have a expert review helper tool, which also gives you evidence that I'll be talking about in the upcoming slides for doing the expert review, and that is called Consolidator. So that's also used for that tool is also used for searching for analogs. That is a very important ingredient or a part of an expert review process. And finally, to generate reports, we have a tool everywhere in Case Ultra for generating different types of reports in different file format like Excel, PDF, Microsoft Word, and so on. So these are the basic ingredients that we supply as part of our software package. One important thing that I need to mention here is that all of these are not different software. They are all combined very nicely into one software, that is Case Ultra. So now let's start with some case studies, and I have few examples that I'll try to go through. Let's see how that goes. So let's take this chemical as an first example, 1,2-epoxy-dodecane. And the first thing when we are trying to determine is to check if we have any experimental data for this compound or not. And it happens to be that yes, we have, and this compound is experimentally Ames negative in OECD 471 strains with or without S9 activation. So that's a very important observation, and right away we can reach to our final decision, which expert decision. That is, it's a class 5 compound based on experimental data. It's an known Ames negative compound. Now we don't have to do anything after this. We are done. We got our expert decision, final outcome, just by looking at the experimental data, which is adequate, looks like at least. But let's see what else is present for this chemical from the software as far as the evidence is concerned. We don't need that, but still let's look at it. So if any identified alerts are found, yes, there is one alert that was identified, which is an epoxide, and which is highlighted in this chemical. Now let's find out if this alert is really relevant or not. Again, we do not need to do that because we have already decided that it's a class 5 based on the experimental data. But let's do just for our curiosity. So how to find out if this alert is relevant or not? We can check analogs for this alert, and here are some of the analogs, as you can see here, and these analogs this is the alert, by the way, and these are the analogs that contain the same alert, epoxide group, and have the same type of environment around the alert, long chain, and every one of them is Ames negative. That means they are experimentally Ames negative compound. So looks like this alert is actually non-mutagenic. Now, if there is any mitigating feature, so mitigating features are those part of the chemical that actually inhibit the mutagenicity of alert. So in this case, we have an expert rule mitigating feature, which is actually the epoxide with long alkyl chains, and they limit the availability of such epoxide at the site of toxicity. So we have a mitigating feature. So now let's look at what is the individual model outcome that we obtained. So in this case, the statistical model gave a positive outcome and the expert rule based model gave a non-negative outcome. But as you might have noticed that I have shown this model outcome at the very last step. We did not even need these model outcome at the beginning because we already got the experimental data. Okay, so that's how we went from one evidence to another evidence in a particular order to get our decision. Now, how did you get these analogs? So as I mentioned before, Consolidator is a tool that is integrated in Case Ultra program, which actually helps us to get these analogs. Okay, so now let's go to the next case study. So this is one for cineol, and let's start with the same process. Let's first see if there is an experimental data available for this compound. Looks like there is no data available, at least we don't have this data. And let's go to the next piece of evidence. Are there any alert that we identified by processing this with QSAR software, statistical as well as alert based, expert rule based? No alerts were identified. So that's it. We can reach our decision that it's a class 5 compound, but let's look at some closely related structure analog to reinforce our decision. So here are some analogs that we found from the Consolidator, and looks like they all have same type of functional groups and they are all Ames negative. So with that in our hand, we can actually arrive at our expert decision that is a class 5 compound. It's negative based on no structural alerts identified situation, and also the decision is supported by analogs, related analogs. And just as a case, these are the default model outcomes. From statistical model we got a negative outcome, and expert rule based we got a negative outcome. So here is our second compound. Let's go to another one, methyl isoeugenol, and do the same thing. Experimental data, no experimental data is available for this compound. If we look into the identified alerts after performing the QSAR predictions, we did not get any expert alert, but we got 2 statistical alerts, which are highlighted in the red part of this molecule. So that highlighted area actually contains two very closely related statistical alerts. So we got some alerts when we perform the QSAR evaluation from the default models. And now let's find out if this alert is relevant or not. Let's try to validate the mutagenicity potential of this alert, and let's see. So these are the analogs from Consolidator, which contain the exact same alert in somewhat same environment. And as you can see, all these 4 analogs are all experimentally Ames negative compounds. So on that basis, we can think that this alert is not relevant and it's not mutagenic. So the expert decision, class 5, negative. The alerts are dismissed using analog data. That's what we did. So we reached to our conclusion. Now let's look at the default model predictions. Statistical system gave us inconclusive, and the expert rule based gave us negative. So as you probably already have noticed that I'm actually giving the default model outcomes at the last, just to emphasize the fact that in many cases you do not even have to pay much attention to the model outcomes. What is important is the evidence or the reasons of toxicity, which are alerts from the models. That are the main important factors in the expert decision making process. Okay, another example. Here it's a little bit larger molecule, and no experimental data is available for this compound. When we looked into the QSAR predictions, we got 2 expert alerts and 3 statistical alerts identified, which is highlighted in this molecule. And as you can see, there is a peroxide functionality on the left side of the molecule, and there is a acetate group on the right side. And let's find out if these alerts are valid or not. And let's take one alert at a time. First, let's check the validity of the peroxide alerts. And here we got two analogs from consolidator. Both are Ames positive. And as you can see, that means that this alert is actually looks like mutagenic, having a good deal of mutagenic potential. Now let's look at the other alert, acetate group, by using the same process of finding analogs. And as you can see, we got two analogs, both are Ames negative. So one alert is positive and one alert is negative. I mean, one alert is valid and another alert is invalid. But it is only one alert required to create mutagenic potential. So it looks like this compound is going to be mutagenic. So the expert decision is class 3, positive and based on valid structural alert, which is the peroxide group. And since we got expert alerts, so we know the mechanism, and it is further validated by using analogs from the database. So that's our expert decision, class 3. Now, mitigating feature, there are some mitigating features are present, not required in this decision making process, and we also found out that they are not related to the alerts, so they are not really meaningful. And default model outcomes, as you can see here, we got actually contradictory outcome, negative from statistical system, and expert rule-based system gave us positive. But we already did our analysis and we found that there are valid alerts and it's a class 3 compound, most likely. Okay, let's take another one. So this is phenyl glycidyl is ester, and experimental data, let's look at it. Yes, there is experimental data, and looks like this experimental data is very strong mutagenicity, Ames positive from different strains, and it is also carcinogenic in rodent assay. And in order to get that, we have used our databases that I indicated before. We have mutagenicity database and carcinogenicity database, which actually not only gives us the experimental outcome, but also tells us what kind of strains are used, and the experimental conditions too. So we have experimental data for this compound, and that's why we can immediately reach to a decision. It's a class 1 compound because it is known mutagenic and known carcinogenic based on experimental data. If we look into alerts, we got 2 expert alerts, 1 statistical alert. Again, to emphasize, we don't need them, but just as a additional interesting factor, I'm showing them because we already achieved our expert decision. So 2 expert alerts, and basically all these alerts are related to epoxide functionality. And if we want to find out analogs for it, here are the analogs, which contain exactly the same group in the same environment, and all are Ames positive. Now please take a step back and try to remember the previous slide where I showed the same type of epoxide group, but in that case it was the analogs are negative. Why? Because in the previous case, the epoxide group was attached with a long alkyl chain, and in this case the epoxide group is attached with a different type of functionality, and that actually proves that the same alert can be positive in some cases and negative in some other cases based on the structural environment around the alert. So that is why it is important to evaluate the alerts based on analogs, closely related analogs. And let's see the default model outcomes, known positive from both, which is expected, and this is a class 1 compound. Okay, so let's take another one. I really apologize for showing so many examples, but I tried to gather examples that show different types of expert review. No experimental data is available for this compound. When we checked for alerts, we found no expert alerts, but the statistical methodology gave two alerts, and that is highlighted on this chemical. Now let's find out analogs. These are analogs which contain the exact same alerts, and they are all positive. So looks like that it's a positive compound. So our expert decision is class 3, but I put a question mark near it, and I'll talk about it later. But it's on a very surface analysis, it looks like a class 3 compound because of valid structural alert and supporting analogs. But when I looked into the metabolism of this compound, because I found out that the acetals are actually converted, they are unstable, they converted to aldehydes inside body, and that aldehyde is the responsible factor for causing mutagenicity. But in this case, the metabolite, the aldehyde metabolite, is a known Ames negative compound. So now that's why I put this question mark. So it could be a class 5 compound based on this data. But since I do not have enough expertise to confirmly to tell that, you know, this Ames negative compound is all what will happen with this parent compound. You know, there might be some unchanged compound remaining which might cause toxicity. So I don't know enough about it. So that's why I put a question mark. So sometimes we have to take into account the metabolism data also. Mitigating feature, there are some mitigating feature obtained, but they are not relevant. And the default model outcomes were inconclusive from statistical system and negative from the expert rule-based system. But, you know, on a first cycle, we got class 3 judgment. So this one is a small chemical, dimethyl sulfide, simple chemical. We got no experimental data for it. And the alerts there were no alerts, and the default model outcomes is out of domain from both the methodologies, statistical as well as expert rule-based system. Why I'm showing this model outcomes at a earlier stage here, because in this case they are out of domain. That's why. And there are uncovered features, and that is why the out of domain outcome were received, and we call it as unknown fragment. And in this case, the whole molecule is actually unknown. That means there are no chemicals in the model training set which actually contain this type of chemical fragments. So that's why they are called unknown fragments. In consolidator, we can check if this fragment is present in any other known chemical that are not present in the training set. And in this case, we found 2 chemicals which contains very similar fragment, and both are Ames negative. So on that basis, we can reasonably say that this fragment may not have any mutagenic potential. So on that basis, we can achieve or arrive at a class 5 decision: negative, unknown fragments deemed to be unreactive. Okay, so those are all our expert review case studies. Now let's look into the issue of how expert review process can serve as a shock absorber for different software versions. So in this case, I'm showing an example where we have used 2 different versions of Case Ultra. Version number 1705, which was, as far as I remember, 2 years ago, and 1805, which is the latest version. And as you can see, the statistical system was negative before, and now it is positive after the version change. But the expert rule-based system remained the same, negative in both the cases. So that's actually majority of the time that's what happens. The statistical system is the one which changes from one version to another. And in the latest version, it also identified 3 statistical alerts, which are in combination highlighted in this case. But if we check out the analogs of this alert, considering that we have seen them and we got a positive outcome from statistical system, we'll see that all this alert, when present in experimentally known compounds with experimentally known Ames data, they are all negative. We got 3 analogs. They contain these different statistical alerts, and all of them are negative. So on that basis, we can call that we can say that it's a class 5 compound. So even if we have changed the version number from one system to another, we can perform an expert review, and most of the times you will see that even the default model outcomes change from one version to version. The expert review will lead you to the same conclusion. So that's why I termed it as a shock absorber. So the expert review process is a good buffer that prevents you from getting exposed to different call changes from one system to another. So finally, this is a class 5 compound. So now, since we have done with our expert review case studies, let's talk a little bit about what are the acceptability criteria of experimental data. I'm sure majority of you already know this, but the AIMS data that if we use in our expert review should have some minimum criteria. That is, they have to be conducted using OECD 471 guideline for both AT and CG type of mutations. So Salmonella typhimurium has these guideline based strains that should be used, and the E. coli WP2 strain or Salmonella typhimurium TA102 has to be used. And these strains should be used both in the presence and absence of proper metabolic activation or S9 system. The concentration of the test substance should also meet minimum requirements. And if carcinogen data is used for determining class 1 compounds, they also should be robust enough according to these guidelines. I'm not very knowledgeable about them, but these are the basic criteria of experimental data. Also, experimental data would actually do have different criteria for positive and negative compounds. In order to call something negative, it has to be negative in all the 5 strains if we are talking about experimental data, with and without metabolic activation. But for being a positive, only thing that is needed is positive in at least one strain with or without metabolic activation. So being conservative, the criteria for being positive is low, but in order to call something experimentally negative, we gotta raise our bar. So in summary, few things we can say. That is, the expert review process for computational assessment of DNA reactivity is an essential step. It is the process of review and verification of the QSAR algorithmic and computational procedure by a human expert with proper knowledge. It can very well be considered as a third methodology in addition to the statistical and expert rule based methodology that the guideline talks about. So there are now 3 methodologies in effect: statistical, expert rule based, and expert review process. And also, it is not an automated process. It has to be done by a human expert. However, the software tools can help in great deal to increase the effectiveness of such review process and to perform it faster. So that's my summary. So with that, I conclude, and I'd like to thank you very, very much for attending this webinar, and thanks for giving us your attention and time. In case of any question, please don't hesitate to email us in the email address that I just gave. And once again, thank you very much. Have a good day.